Assalamu alaikum, and good evening, everyone. Thank you for the introduction. I was talking about my work to Dr. Zafar Bashir. Previously, last 5 to 10 years, my lab is involved in the identification of different compounds that is being isolated from different extracts that have ability to reverse the neuroinflammation or neurodemyelination, or have ability to increase neurite outgrowth from the neurons. Because neurite outgrowth is the process which is extremely important during the development. Because when you have an inflammation, because inflammation leads to the development of demyelination, they also lead to the destruction of the neuronal cells, as well as they disrupt the signal transduction pathway, as well as disturb the neural impulses from the neurons. So my lab always in search for different compounds that have ability to reverse the neuroinflammation or neurodemyelination in the brain. Because if you overcome the neuromyelination, you can overcome the demyelination, and it is also very beneficial. And molecules that that help in protecting the neurons under inflammation as a neuroprotectant. So previously, my lab works with different isolated and purified compounds. We have isolated and purified compounds and characterized the compounds by different techniques like LC-MS, NMR for the characterization. And we have isolated previously, and research papers have been published that deals with medicacic acid, gallic acid, vanillic acid, and piperine. So we have already established that medicacic acid, which is isolated from herbal extract, has a potent effect on reducing the fast transient potassium channels, and it promotes neurite elongation in the hippocampal CA1 neuron. And by doing that, what happens in medicacic acid during the development, it internalizes the fast transient potassium channels, thereby helps in the increase in the neurite outgrowth from the hippocampal neuron. Whereas piperine, we have isolated piperine from the piper, black pepper, and we show that it has ability to inhibit cancerous activity in the neuroblastoma cell lines, that is the cell lines of which is present in the young children. And we show that piperine, along with other extracellular matrix proteins, not only CSPGs, that are chondroitin sulfate proteoglycan, which is very non-permissive proteins, but tenascin C, D domains, and tenascin C is also very important protein besides chondroitin sulfate proteoglycan that are required to inhibit cancerous activity in the neuro-2A cell line. Besides that, we have already established gallic and vanillic acid. This is also two very important acylated compounds from the herbal extracts that shows that it also has ability to inhibit inflammation and neurodemyelination in a neuron glial ex vivo model of neurodegeneration. So to extend the previous work, we have isolated again a very interesting and novel compound that is called as protocatechuic acid. Basically, protocatechuic acid is also called as 3,4-dihydroxybenzoic acid that belongs to non-flavonoid class of polyphenolic acid. It is present abundantly in edible food such as strawberries, honey, raspberry, white grapes. It is present abundantly in those food products. And its structure is, as it is 3-hydroxybenzoic acid, and it's a phenolic acid phenol compound. So its structures has similar structure with other antioxidant molecules that has been identified and previously described. So in order to visualize the effect of protocatechuic acid in neurodemyelination or in the reversal of neuroinflammation, or whether just to see that how much has this protocatechuic acid has effect in reversing neuroinflammation and neurodemyelination, first we have to prepare a model of neurodemyelination. So we have prepared our ex vivo model of neurodemyelination, where we have co-culture neuroglia. Where we have oligodendrocytes, we have neurofilaments, we have astrocytes, and we have microglia cells as well. if you see here the picture which is present at the left-hand side, you can clearly see the MBP positive or myelin basic positive oligodendrocytes along with the neurofilament stained characterized protein of neurons and neurites. Neurite and DAPI is used to just to stain the cells, because in the vicinity you have astrocytes and microglial cells also present, which are obviously not positive for MBP and neurofilament. So just to show you, at the right hand side you can clearly see that the neurons with the long axons and neurites of hippocampal neurons. You can also see oligodendrocytes, a star-shaped small cell body, but star-shaped oligodendrocytes with lots of branches and processes, and flat-shaped large cell body structure that is called as astrocytes. They have a ciliate-like structures and are present abundantly, and microglia are also present in the co-culture. So first we have to prepare the neuroglia co-culture, and then we use lysolecithin to induce demyelination in the co-culture and induce neuroinflammation. So what will happen when we add lysolecithin? You all know lysolecithin, it's actually solubilize the lipid bilayer and causes the release activation of phospholipase A2 from the macrophages. That cleaves the phosphatidylcholine into lysolecithin and then converted into lysophosphatidic acid. And then it leads to the causes the inflammation and demyelination. So we will check it in the next round of experiments. Now this is the experimental protocol, how we do it. So first we have isolated the neuroglia co-culture. So what we did, we actually cultured them like 2 weeks after from the brain dissection. We cultured them glia for 2 weeks, and after, when they become confluent, when they become confluent, we coat them or culture them in poly-L-lysine or poly-L-lysine amino acid coated cover slip. And after 24 hours of 2 days of culture, we add hippocampal neurons on them. So monolayer of hippocampal neurons being cultured with co-cultured with the glial cells that contains oligodendrocytes, astrocytes, and microglia. And after 5 days in vitro, we have added ascorbic acid. Ascorbic acid is being added just to initiate the myelination process. But if you didn't add ascorbic acid, it will take longer time for the myelination. But in order to increase the rate of myelination in vitro, you add ascorbic acid. So after the addition of ascorbic acid at day 11, one group acts as a control, the other group receives lysolecithin. Now, lysolecithin is being used, and specifically this concentration, 0.003% concentration, is used as such that it leads to the minimal cell loss and cell death. Not all cell died, or not all myelination or all the neurons or neurites get demyelinated. So very critically analysis takes place in order to check and to calculate the percentage of lysolecithin that is being used in this culture. Afterwards, we checked whether our model of neurodemyelination is developed or not. So in order to check, we used immunostaining and patch clamp experiment. Patch clamp experiment has been used to identify, to measure sustained repetitive firing. And we have to use, because obviously when the demyelination takes place, neural impulses alteration takes place, sodium potassium channels are disturbed. So obviously, oscillation, sodium channels, membrane potential changes. And immunostaining is used by using different antibodies. We will see what are those antibodies in later slides. And then afterwards, at day 12, again, the lysolecithin, after addition like 15 to 18 hours, lysolecithin is being removed and it is replaced by fresh media. So this group also divided into 3 groups. So first group received fresh media, other group received fresh media along with the compounds, protocatechuic acid. And previously we have added gallic acid and vanillic acid, but in this study we use protocatechuic acid. And then after 2 days, or day 13 or day 14, we use again immunostaining and patch clamp. Sometimes patch clamp also analysis takes place at day in 15 days after in vitro. So this is actually how the experimental protocol takes place. It takes very a lot of time to optimize this experiment. Now, first and foremost, we need to see how much has PCA or our compound has capability to initiate or to activate or to increase neurite outgrowth from hippocampal neurons. So what we did we actually culture hippocampal neurons. We dissected out hippocampus, and then after processing it, we cultured the hippocampal neurons on poly-L-lysine coated cover slips, and we have to select dose concentration in order to for the further experiment, because we didn't know that which dose can produce submaximal response. So we need to identify, we need to check that to select doses which produces submaximal response. So we used a lesser amount like 2 micromolar, 5, 10, 25, 50, 70, and 100 micromolar doses of protocatechuic acid. If you see the structure you see, and we measured the neurite outgrowth by a specific software which is called as morphometry, and we traced down the axons from the soma and towards the terminal tip of the axons or the neurites. And we measured it by a specific morphometry software. And after the measurement, we use 2 parameters of neurite outgrowth. One is the mean average length of the 10 longest neurite, as well as we have identified sum of all the neurite lengths. Now, just to increase the mean average length of the 10 longest neurites, the reason is that we need to know that how much PCA has ability to extend or to stimulate long neurites, long neurites. And the sum of neurite length is just to give the approximate efficacy of protocatechuic acid, that how much of the sprouting and how much in total it increases the neurite outgrowth. So if you see this graph plotted against the concentration and the length of the neurite outgrowth in micrometers, if you see here, there is a dose-dependent increase in the neurite outgrowth takes place. It increases the sum of all the neurite length as well as the mean average length of the 10 longest neurite. And the doses that were selected that produce the submaximal response is 10 and 25 micromolar. Concentration has been used and been selected for further experiment in this study. As I told you that.
Sorry, Sonia, could I just ask a quick?
Yeah, sorry.
Can I just ask a quick question, please?
Yeah, sure.
Yes, in the previous slide, how long is the PCA applied for each concentration?
How much or how many days?
Yes, how many days
2
is
days.
it?
It's 2 days. It's 2
2
days.
days for, and this is sequential
Yeah, 2 days.
application, is it?
Which one? Sorry. Okay. Sorry. No. Sorry. In this diagram, no. It's 24 hours or 48 hours. But this is from the 24 hours. 24 hours. Yes.
Right.
This one is 24 hours.
And so each concentration is applied for 24 hours, or the whole 24 hours?
Yes. Yes. No. Each individual. We coated these, cover slips. We have cover slips, 10 millimeters of cover slips, and we coat them cover slips with with poly-L-lysine, and we added concentration, each individual concentration on individual cover slips. Yes. 24 hours. Now, when we calculate this one. Then what we did, we have prepared neuroglia co-culture, as I told you. So if you see here, the co-culture in the neurons, specific antibodies, which is the neurofilament, and myelin basic protein is used to characterize oligodendrocytes in the culture. So if you see here the control region in the control picture, the co-expression of myelin along with neurofilament is clearly seen. Where just see here, and other the only red one that is being present like oligodendrocyte is being also shown here and here. So co-expression of neurofilament and MBP is there. That shows that myelination is there, and we can proceed with the experiment. Then afterwards, in this neuroglia culture, we added lysolecithin. So just see here, this is the figure in the lysolecithin. Just see the morphology changes suddenly. The course, the length of the neurite length, and the expression also decreases. Whereas MVP also, the length in the co-expression, just see the expression of neurofilament and the expression of myelin basic protein also decreases from the control level. The neurite length also decreases, as well as the expression also decreases. As I mentioned, that that we selected lysolecithin concentration as such that it didn't produce a lot of cell damage, and not all the demyelination takes place at once. So we also checked through image software the mean fluorescent intensity, and we have identified that that after the induction of LPC, the expression of myelin basic protein as well as neurofilament is decreased significantly from the control levels. So this is just, I think I skipped one. It's just okay. In second, we used to see GFAP. You all know GFAP because GFAP is a specialized characterized antibody to identify astrocytes. And we use the co-culture, and we use the co-expression to see the co-expression of neurofilament with GFAP. After the induction or after the treatment with LPC, the neurofilament, obviously, the expression is decreased, whereas the hypertrophy of astrocyte can be clearly seen. The astrocyte morphology is T-like structure, spongy-like structure, but they are become hypertrophic because when the neuroinflammation takes place, GFAP it becomes reactive astrocytes, and the reactive gliolysis is induced by the activation and increased levels of ciliary neurotrophic factors as well as interleukin 1 beta. And then if you see here, the high expression of GFAP and the low expression of neurofilament can clearly see after the induction of LPC. We also use chondroitin sulfate proteoglycan as well as tenascin C expression. As you all know, chondroitin sulfate, here we use specific antibody to identify chondroitin sulfate 56 stub. So whether it is dermatan sulfate, keratin sulfate, or heparin sulfate present, it all recognize, that antibody recognize all the chondroitin sulfate 56 stub present on all the proteoglycan present in the oligodendrocyte, or the astrocytes, or in the neuron. We also use laminin. Laminin is the basement membrane protein, is also extracellular matrix protein, and it is a basement membrane protein, and neurons loves to grow on them, and they are more permissive molecule than the chondroitin sulfate proteoglycan. Because chondroitin sulfate proteoglycan, they are extremely non-permissive molecule, and when during the development, these proteins expresses themselves in a very high concentration along with their receptor beta zeta receptors, and they have involved in migration, proliferation, and differentiation. But when the development of brain inhibitor stops, or after the maturation, when their expression and their receptor, the expression of these proteins and their receptors expression, they goes down. And when there is an insult of the brain, and when there is an inflammation or an injury in the brain, the levels start to increase again. That corresponds to the neuropathological pathological state of the brain. So we use laminin. So here, if you see the co-expression of chondroitin sulfate and laminin in the control region, so you can see differential expression of both the protein in different cells, because our neuroglia culture has not one type of cell, but they contain 3-4 different type of cells. So each cell expresses different amount of proteins. So therefore, we have checked the amount of these proteins by STS page electrophoresis. I'll show you in next few slides. But just to see here, the co-expression of laminin and chondroitin sulfate, which is present here. After the induction of LPC, the expression of chondroitin sulfate becomes way more, increased in a very concentration, twice as high as in the control areas, control cells, as well as the concentration or the expression of laminin also significantly increased from the control cells. Both and this is the laminin increase in the expression of laminin just act as a SOS response from the cells, because when the non-permissive molecules are increasing in the level, increasing after the neuroinflammation, the permissive molecule expression also increases just to neutralize the non-permissive milieu. Unfortunately, at certain point, after a while, laminin levels remain the same, but the rising of chondroitin sulfate proteoglycan simultaneously, continuously in the brain leads to more destruction and leads to the formation of clear scar over which no neurite can form and can regrow on them, and it inhibits neurite or synaptic connection. So if you see here, the co-expression of chondroitin sulfate proteoglycan and laminin after the LPC induction, both the concentration, the levels, or the mean fluorescence intensity of laminin as well as chondroitin sulfate is increased, significantly increased from the control cultures. We also use tenascin C, and we see because the tenascin C is also an extracellular matrix protein, very important. Tenascin is a protein, and it has 5 different splice variants: tenascin C, R, W, Y, and X. They all are involved in neurite outgrowth, in neurite modulation, adheration, neurite guidance, proliferation, differentiation. And tenascin C specifically, it's very important because it's a hexabranchian. This tenascin C and laminin also compete for the same receptor, beta integrin. It also has different receptors like RF11 receptors, and contactin receptors are also there. So during development, similarly, like CSPG, tenascin C also increases in a very high amount during development. But when the brain development decreases, or it stopped or halted after the maturation, its levels remain, its levels start to decline, as well as its receptors start to decline. But once there is a brain injury, the level becomes increased. So they all are, these molecules has been used in our study just because of these speciality, because they all are increased during the development. And these are the molecules which are extremely non-permissive and are present in the glia, and they form gliotic scar over which neurons cannot grow. So if you see here, as this is extracellular matrix protein, so its concentration, its expression can be seen at the periphery, not in the inside of the cell. But when there is, when LPC is being added, if you see here, the aggression or aggressiveness or the high expression of tenascin C can clearly seen in LPC induced model of neuroinflammation. The levels or mean intensity levels are becomes twice as high as from the control regions. Now we also use check the expression of nuclear factor kappa B and the cyclooxygenase enzyme 2. Now we have in cyclooxygenase enzyme 2 and cyclooxygenase enzyme 1. One is constitutively expressed, therefore its expression we didn't see because its expression remains the same. However, we use to see cyclooxygenase level, cyclooxygenase 2 levels, because it is inducible by neuroinflammation. And when there is a stress, when there is inflammation, COX2 and NF-kappa B levels increases, and they lead to the activation of mitogen activated protein kinases as well. So we check the nuclear factor kappa B. Here we use antibody that recognizes the nuclear local signal. We have two heteromer, two dimer, it's subunit P50 and P65. We have used here to identify NF-kappa-B, which recognize P65. Now, P65 is very important because it is activated and involved in stress-related processes and increase the level of reactive oxygen species. So therefore, we have used. So you see, after the induction, after the LPC treatment, its expression from the center, because in the quiescent state, NF-kappa-B remain inside the remain out in the periphery. But when it becomes activated, right, by kinases or by kinases, then it translocates itself from the periphery to the interior of the cell. So if you can see here, the morphology of the cell changes and expression level from the periphery to the center when the cells are treated with lysolecithin. Similarly, is the case with the COX-2 proteins. Then we have used, because the pure or number of sodium channels with slow sodium dependent membrane potential oscillation are also disturbed when the myelin is degraded, which produces ectopic sodium oscillations along the neurons. So here we use, during the patch clamp experiment, we used to measure latency period, number of spikes, resting membrane potential, and amplitude of the signal. This is pyramidal hippocampal neuron, and this is a pipette, very thin pipette. The diameter of the tip of the pipette is 2 to 10 mega ohm, which is very important in order to generate very high strong giga ohm field. And the pipette is filled with ICF, which is relatively contains lots of amount of potassium as comparative to ECF. So the cells are present in the ECF, and the pipette contains intracellular fluid. Now, the patch clamp is a very sophisticated technique through which we can identify, record current that is passing through single ion channels. Either we can record the membrane potential or sustained repetitive firing, or we can identify the current. We identify current on specific membrane voltage. So here, in this diagram, this is whole cell recording, just to show you that this is the cell membrane, and this is the glass pipette, borosilicate glass pipette. So we make, so for first and foremost, we make a low resistance seal with 50 mega ohm. Then a little suction is being applied, so a little membrane pouch inside the pipette. We and make a 10 to 100 giga ohm seal. Now, the more the resistance it shows, the more better current can be recorded, and there will be no leaking current is there. Because if there is a leaking current from here or here, because if the giga ohm seal is not tight enough, so the leaking current from here and here can not give you exact measurement of the firing or the current that is passing from this channel that are present here. Now, after applying a little more suction, which disrupts or disturbs or destroy or break the patch. This is the patch, so this is the patch, right? So therefore, it is called as patch clamp. So make a little patch. Now, the current that's passing through all the cells is now recorded from the pipette, and then we can record the current. Now, it has two modes. One is current clamp mode, and the other one is the voltage clamp mode. So in the voltage clamp mode, what will happen? You actually hold the voltage. Means, for example, you hold the voltage. For example, if you hold or control the voltage at minus 55 millivolts, and you then applied current from minus 60 millivolts to plus 60 millivolts with 10 millivolts of interval. So you can check that at the minus 55 millivolts, how much current or from which ion channels are opening and producing what type of current, and how much current is being produced when one channel is open or not at a specific giving membrane potential. In current clamp mode, you applied, as a term suggested, current clamp. So if you here, you actually hold the current and you see the oscillations or membrane potential, see, in the form of spikes. This is also called as sustained repeated firing of the neurons, from the neurons. So here in this experiment, we use neurons that has minus 55 millivolts of the holding potential or more than minus 55 millivolts of membrane potential. And we applied 50 picoampere current for 400 milliseconds. When we applied this current for 400 milliseconds, the neurons get excited and fire action potential or fire sustained repeated firing. And the amplitude of the firing, the spikes, or the number of the spikes, the amplitude and the frequency can all be measured. So molecules that are good to inhibit sustained repetitive firing act as a good anti-epileptic compound. Okay, so here we also, as the demyelination takes place and neuroinflammation takes place, so obviously the oscillations are changes, sodium dependent oscillation, membrane potential changes. So what we did, this is the co-culture, neuroglide co-culture system. This is LPC, after LPC, and this is in control situation. So in control situations, they all have a sustained repetitive firing. In LPC, we have identified that there are less number of firing than in control. And the important thing is that to note that the latency period means latency period is delayed, means in the cultures or the neurons that are present in the LPC after the treatment with LPC it takes a lot of time in order to initiate or to initiate first action potential. So the latency time to initiate an action potential is actually delayed when you compare it from the control. And the number of spikes are also delayed or also decreased from the control situation. Other features of action potential or other measurements that we have used, like for example, threshold potential, action potential duration, and amplitude of the spikes can all be measured, but we didn't find any significant differences between the control and the LPC treated cultures. So at this point, we have identified that the neurite that our neuro model of neuroglia model of demyelination or neuroinflammation is successfully developed. Now we are ready to apply our compounds that we have selected, and we see whether the compound has ability to reverse the myelination process, reverse the sustained repetitive firing, or reverse the high expression of reactive high expression of ECM proteins and pro-inflammatory markers in the reactive astrocytes and reactive glial cells. We used another antibody that specifically recognized neurites and neurons, that is beta-3 tubulin. And we use it here. If you see here, beta-3 tubulin is much more nice than the neurofilament. And after the LPC treatment, the beta-3 tubulin expression becomes decreased, whereas after the addition of protocatechuic acid, the expression and the neurite outgrowth has increased. So the mean average length of the 10 longest neurite after the addition of protocatechuic acid, both the concentration increased the mean average length of the 10 longest neurites, as well as the sum of all the neurite lengths has been increased. We also check whether low expression of myelin and high expression of GFAP or contractile sulfate proteoglycan and senescence C is decreased by the presence of protocatechuic acid or not. So we again applied after this is the co-culture in the control situation, and when we add protocatechuic acid, the levels of MBP. The levels of MBP or the co-expression of MBP on the neurites is increased, and you can clearly compare it to the control situation. The co-expression of neurofilament with the MBP at a higher concentration of protocatechuic acid as well as the low concentration is also can be correlated with the control situation. And you can also see the expression here, which is not very good, and there is very less co-expression of myelin, and neurofilament takes place. So punctate structure and coarse neurite can easily be seen in neurite in after LPC treatment. The mean average fluorescent intensity also shows the same, that it increase after the addition of compounds, it increases the expression of MBB and neurofilament. So we again check the expression of GFAP, and in the co-culture system, you see if you compare it, the co-expression of of neurofilament with the glial fibrillary acidic protein, and in the LPC treated and after the addition of protocatechuic acid, it increases the neurofilament expression, the neurite outgrowth, as well as the expression of GFAP becomes down-regulated, and you can easily compare it from the control situation. If you compare it from here, see here, it becomes hypertrophied and very very stellate structure and bushy structures are there, which become reactive gliosis. But here, the expression and the morphology is not remain hypertrophied, it remain and looks like in the control situation. The mean fluorescence intensity of GFAP and neurofilament is also altered by the addition of protocatechuic acid. Then we have checked the expression of laminin. Laminin expression in LPC after a few days, it remains high, but after a while, the continuous synthesis of chondroitin sulfate proteoglycan makes the environment so inhibitory that laminin concentration, which halted after few days, few hours of injury, is unable to repair or unable to make the non-permissive milieu into permissive one. So when we add protocatechuic acid, the expression of chondroitin sulfate proteoglycan as well as here this is chondroitin sulfate proteoglycan and laminin. Sorry, this one is the laminin one. It becomes or remains becomes normalized if you compare it from the control situation. So this is the co-expression of CSPGs with laminin, and this is you can clearly see in low concentration and as well as in the higher concentration after the addition of higher concentration doses of protocatechuic acid. We also checked the level of other proteins, tenascin C. Okay, tenascin C. The expression of tenascin C also shows the same. The higher expression of tenascin C becomes down-regulated, and the pictures can clearly see and correlated with the control situation. Even though the punctate expression of tenascin C and the expression at the periphery can be clearly seen in both the doses. So both the doses decreases the higher expression of tenascin C. At the mean average fluorescence intensity is also decreased when the compounds is being added. NF-kappa B, it is a nuclear factor kappa B, very important pro-inflammatory marker, which is in the quiescent state, remains in at the outside, but once it is activated, then it becomes to translocate itself inside the nucleus. And after the addition of the compound, both the compound, the morphology of the cell and the staining and the expression of NF-kappa B retains towards the normal concentration, normal level. The COX-2 cyclooxygenase enzyme levels also shows the same expression. If you compare it from the control level, the COX-2 levels are significantly decreased, the mean fluorescent intensity after the addition of protocatechuic acid. So we also see that we also measure and we also identify and investigated the effect of protocatechuic acid in the downstream pathway, that how it actually involves in the signal transduction pathway. because normally we know that the phosphorylation of these molecules, because the external stimuli, what will happen when there is an inflammation in the brain, the phosphorylation of different downstream proteins takes place. And when we talk about neuroinflammation, we talk about NF-kappa B and cyclooxygenase enzymes. They both are phosphorylates and activates mitogen activated protein kinases and activation of protein kinase C as well. Now, protein kinase C also activates by the inflammation when the diacyl glycerol kinases are present in a larger amount in the cell. And it also increases the oxidative stress, and the production of extracellular matrix also takes place. Normally, the phosphorylation by the external stimuli, when it is in oxidative stress, it increases inflammation, leads to the phosphorylation of protein. Besides PKC, extracellular receptor kinases and mitogen activated protein kinases also take place, which activates cyclic response element binding protein, transcription factors that activate cyclic response elements, and they bind at the nucleus, translocate themselves into the nucleus, and bind at the and on the DNA, at the DNA where the RNA polymerases bind. So when the RNA polymerases bind and it activates, it initiates the transcription of the structural genes. And here in this case, the structural genes are increased expression of inhibitory nitric oxide synthetase, increased expression of tumor necrosis factor, increased expression of prostaglandin, increased expression of non-permissive proteins such as contractin substrate proteoglycan, NF-kappa B, and tennyson, and COX-2, and increased amount of ATP production. They all lead to the neuroinflammation and neurodemyelination. So we also, therefore, this is very important. We identify the expression of mitogen activated protein kinases, and we want to see whether protocatechuic acid exerts its effect signal transduction through these pathways. So we checked and we have identified that the mitogen activated protein kinases expression is high when in the LPC treated cultures, then in the control ones, as well as when you add protocatechuic acid. When you treat the cells with the protocatechuic acid, the level becomes towards the normal control ones. As again, I will wanted to express that the co-culture contains not just 1, 2, 3 different type of cells, and all cells expresses proteins differently. Okay, and they have a differential expression levels in all these proteins. So if you didn't see the expression of MAP kinases here, so probably it doesn't mean that it's not increased, but it is increased. Therefore, we did STS page and electrophoresis and Western blotting just to see how much increase in these signal transduction pathway proteins is there. So the mean average fluorescence intensity of mitogen activated protein kinases were quite high when the cultures were treated with LPC, whereas when the cultures were treated with protocatechuic acid in higher and lower concentration, both the concentration decreases the mitogen activated protein kinases. I'm guessing it's just because of the decreased phosphorylation. So protocatechuic acid decreases the phosphorylation of these downstream proteins, thereby reducing and helps in the reversal of the neuroinflammation in our system. We also use protein kinase C because protein kinase C alpha is also involved, and because high expression oxidation or inflammation also increases its expression and causes the phosphorylation of these proteins. So we checked the expression. So in the control, there are less expression. In the LPC, the expression becomes quite high. And when we add protein kinase C alpha in higher concentration or in the lower concentration, both the concentration decreases the levels or expression of protein kinase C, then the LPC treated cultures as well as in the control cultures. And see the levels of just wait, give me 1 second. The neurite outgrowth as well as the co-expression of PKC alpha and neurofilament can also seen, and the cultures here in the lower dose as well as in the higher dose resemble similar as seen in the untreated cultures, and the expression is quite low if you compare it from the LPC treated cultures. So mean intensity of protein kinase C alpha is less after the addition of the compound. So we again check the sustained repetitive firing, and this is the control untreated cultures. This is the cultures that we treated with LPC, and these are the cultures in the presence of protocatechuic acid in both the concentrations. So in the previous experiments, we find that there is delayed in the latency period or the delayed in the generation of first action potential. Similarly, in the case that LPC delayed the initiation of action potential, whereas the higher dose is much more effective in generation of the action potential, but whereas the the lower dose of protocatechuic acid still has a delayed, even though it is not that much significant from the untreated cultures, but it is from the, if you compare it from the LPC, obviously it increases, the delay is less. But if you compare it from the PC10 or both the concentrations with each other, so PC25 is much more effective in decreasing or generating the action potential than the untreated cultures as well as the lower dose of protocatechuic acid. The sustained repeated firing is also become normalized in protocatechuic acid at higher doses and in the control, whereas in LPC treated cultures and the sustained repetitive firing is low, as well as the number of spikes are also decreased. Similarly, there are no significant differences were identified in other measures of action potential generation at day 15. Like we have threshold potential, action potential duration, and amplitude. So there were no difference in all parameters that is being identified. As I told you, that in our culture we have neuroglial culture. Each individual cells have different expression patterns. So we lyse the cells. We prepare cell lysates, and we applied the cell lyses on STS page and Western blotting. So STS page is sodium dodecyl sulfate polyacrylamide gel electrophoresis, is a very specialized technique through which we isolate and we separate proteins on the basis of molecular weight. Because when we applied STS page, we applied STS of sodium dodecyl, it gives equal negative charges on the protein. So when all the proteins are negatively charged, then the protein will only separate on the basis of the molecular weight. And by applying Western blotting, we make the antibody antigen reaction with each other. So if you have a Western blot, so it is specifically binds with its corresponding proteins and epitopes that are present, and then we get the band. The result shows, because we want to see that how much of the protein is there. So this is also very important from the immunostaining pictures, and it's the result of this STS page Western blotting correlates the results. In LPC, this is PKC alpha, this is NF kappa B, this is COX2, this is MAP kinases, and this is P38. Now, P38 is also another very important mitogen activating protein of the signal involved in the signal transduction pathways and related to stress and inflammation. So protein kinase C, the band, just see the very high and thick band of LPC, the increased concentration of LPC, increased concentration of protein kinase C alpha after LPC treatment, and when you compare it from the untreated controls. And when you add protocatechuic acid, the levels of protein kinase C decreases. Similarly, the mitogen activated kinases, protein kinases, levels are high, and it decreases when you have protocatechuic acid in higher doses and compared to the control. On the other hand, P38 is present elevated in a very high concentration after APC treatment, whereas if you compare from the untreated control, and when you add protocatechuic acid in higher doses, then you see that the levels are decreased. Besides this, NF-kappa B similar results being shown, and in the COX-2 also shows the higher concentration than the in the presence of compounds. So we can see this is a very nice diagram at the left-hand side. It shows that this is previously reported, but in our system we also see that in our study that the protocatechuic acid does the similar thing. Now, the inflammation, when it is created by the addition of lysolecithin, it increases the reactive oxygen species, and it also increases the NF-kappa B, or activates NF-kappa B, P38, and JNK, which activates that NF-kappa B also activates cyclooxygenase 2, which is and this induces oxidation and inflammation in the system, and it also leads to the increased synthesis of tumor necrosis factor alpha and inhibitory nitric oxide synthase. And when you add protocatechuic acid through the receptors, of course, they activate phosphoinositide 3 kinases, RAS, and protein kinase C. So phosphorylation of these molecules takes place after the inflammation, whereas the dephosphorylation of these molecules leads to the inhibition of these signal transduction pathway, leading to the reversal of inflammation and neurodemyelination in our culture system. So this is my student who really worked hard with me. These are my PhD. These are the two my PhD students. These all are MPhil students that did work with me. He is Faisal, who actually works very nicely with the Petchlem experiments. And this is Priya, whose work I have shown here. So this whole experiment and work is being supported by Higher Education Commission, Pakistan grant, and both departments of neuroscience at Dow University as well as in Panjwani Center. Half of the work has been done at Panjwani Center, and half of the work has been done in the Dow University of Health Sciences. So this is it. Thank you very much. So if